Hello, my name is Matt. I'm a graduate student at Colorado State in the Department of Biomedical Sciences, and we study the development of a brain region that in yourself is about the size of your thumbnail. It's called the hypothalamus. This brain region is very important for many survival-directed behaviors such as eating, sleeping, fight or flight response, temperature regulation, and reproduction. Now if you take a look at this screen, this is a screenshot of what's called the hypothalamic pituitary adrenal axis. So this is a cartoon of a human brain with a sagittal section, so it's split down the middle. And in here at one is the hypothalamus, and there are cells in the hypothalamus that release a chemical or a hormone called corticotropin releasing hormone. This acts on cells in the pituitary gland to cause them to release something called adrenal corticotropin hormone. This ACTH travels through the bloodstream and acts on the adrenal gland, which is just above your kidney, to secrete something called cortisol. That's a chemical that you guys may have heard of before. And this is very important, again, in the stress response, because under a situation where you perceive a threatening environment, you need this cortisol to allow release of energy stores and cause you to have a heightened state of alertness. For many good ethical reasons, we can't study the development of this brain region in humans. Therefore, we study the development of the hypothalamus in mice. This is an adult mouse brain that's been removed from the skull. And this is an embryonic mouse brain on about day, actually that is actually the whole head. That's on embryonic day 17, which would be 2 days before it was born. So one of the main techniques we use to study the development of the hypothalamus is something called fluorescence video microscopy. And later in the semester we'll learn more about this, but it's basically a technique where we can take video of cells as they actually move in the developing hypothalamus. So we've sent these movies to some folks in our mathematics department here at Colorado State, and they've developed a mathematical model to predict the behavior of these cells. And the model that they've come up with is a model of random walk for the cells with a directional pull. And your homework assignment in lab will be directed towards that. We will actually ask you to model out this behavior on your football field. So this is a picture from actually a brain atlas for a mouse, and this is a coronal section, and this would be the middle of the brain, so this is midline, and this whole block is considered the hypothalamus. So there are cells that populate what's called the supraoptic nucleus, and this brain region is important for maintenance of blood pressure. So cells first are born or go through their last mitotic event along the ventricle, and they have to migrate all the way out to take up residence in this supraoptic nucleus. About 1.4 millimeters in a mouse. So what you have to do for this assignment is create a ratio of your size to the size of a neuron, and then calculate how much of a football field you're going to need to use to model the length of migration that those cells have to take to be in the supraoptic nucleus. So I'm going to give you the size of a neuron is about 10 microns or 10 micrometers in diameter. Now it's your job to take your shoulder to shoulder width and call that your diameter, and calculate how far on the football field you guys are going to have to walk to emulate that migration.